Thank you for the opportunity to present this collaboration between Cienzano and the University of Brussels that was using whole genome sequences in combination with epidemiological data to study the spread of low pathogenic avian influenza virus in Belgium. The outbreak occurred in 2019 in Belgium and was diagnosed to be of a subtype H3N1 and of low pathogenicity. And the full genome of the index case indicated that it may have been a spillover from a wild bird population. However, the outbreak spread really fast in a densely populated poultry area, and it showed an atypically high pathogenicity in reproductively active hens, including mortality, lay drop, and even respiratory and nervous symptoms. When adult layers were inoculated with the virus isolate, systemic replication and disease was shown by two independent teams, and follow-up investigations by German colleagues confirmed the neuro and epitheliotropism and indicated an alternative activation mechanism for the hemagglutinin precursor protein by plasminogen recruitment as an explanation of this pathogenicity. The outbreak affected 81 farms in Belgium and 3 in France and occurred in a densely populated poultry area in the west of Belgium mostly, and the containment measurements and the economic impact were more HIPAD-like, although it was a non-notifiable virus by legal definition. We sequenced 104 complete genome sequences from this outbreak, representing more than 80% of the affected farms, and with this we did a time-calibrated phylogenetic reconstruction, which you can see on the left, with the leaves of the tree representing the outbreak samples, so the viruses from the farms, and the squares are representing the intermediate nodes, which are hypothetical deduced common ancestors between the viruses found in the farms. Now, this kind of reconstruction can then be combined with exact geographical coordinates for each of the samples to be mapped on a map with their longitude and latitude, and this is what we call the spatially explicit outbreak reconstruction. And because there is a phylogenetic uncertainty associated with the deduced nodes in these trees, we also have to plot this uncertainty on the map. Now, what is nice about these spatially explicit outbreak reconstructions is that you can consider them as an independent dispersal model for an outbreak. They are not using any epidemiological data, only the genetic data. And this allows you to do hypothesis testing by comparing epidemiological data to this independent dispersal model. And it also allows you to look at local dynamics, to look at how local correlation exists between epidemiological drivers and spread. Here you can see the time-calibrated reconstruction using the whole genome sequences, and we added a color code to the time scale with the index cases in red and evolving towards blue for the latest cases in the outbreak. And when we plot this on a map, we get the spatially explicit phylogeographic reconstruction. And allow me to zoom in to the densely populated poultry area. So now looking at the densely populated poultry area, we can see the origin region here in red and spread mostly in this direction to affect the whole of the densely populated poultry area. Now this spatially explicit reconstruction was used to test specific hypotheses. And the first hypothesis we tested, because this was hinted at by the field veterinarians, was an effect of wind on the dispersal of the virus during the outbreak. We could use 2 meteorological stations with continuous wind data in the densely populated poultry area, providing both wind direction and speed. And when we looked for the whole of the epidemic, whether there was a correlation between dispersal direction and wind direction, we could see that there was a positive but not very strong statistical support for wind having an effect on dispersal in the whole of the outbreak. When we subdivided this in time slices corresponding to the main outbreak management decisions, like for instance a standstill of transportation, we could see that this effect was limited to a specific period in the outbreak, and this is the period when the most of the densely populated poultry area was reached. And so we can conclude for the wind that there is a moderate effect of the wind on the dispersal of this virus during a specific phase of the epidemic. Then we set out to test other hypotheses by looking at other epidemiological variables like geographical proximity, transportation links between farms, common visits by veterinarians, or family links between farms. And we first used univariate tests to explore which epidemiological variables may have an effect on dispersal, and there only geographical distance and visits by the same vet came out as significant. But as you can imagine that there may be a codependency of several of these variables. You could, for instance, imagine vets visiting geographically more closely to each other farms more frequently within a multivariate regression analysis, which indicated that only geographical distance was the main predictor of the epidemiological connection overall for the whole of the outbreak. Now, looking at local signals, we did another kind of analysis where we used the spatially explicit reconstruction as a backbone to check to what degree epidemiologically identified networks would occur on genetically linked farms. And this is something you can do by giving a same color code to an epidemiologically connected network. For instance, here in the middle you could imagine all of these red farms having the same color because they are visited by a same feed delivery truck or another connection. And here for human movements you could imagine all these yellow farms to be operated by members of the same family, or another social connection between farms. When we plot these epidemiologically identified networks on the tree, you can see that often, but not always, they cluster together in genetically related clusters. And this is something that is even supported very significantly by statistical analysis. So in conclusion, we can say that these full influenza genomes, they provide a good resolution to do spatially explicit outbreak reconstruction. And that with this spatially explicit outbreak reconstruction, we have an independent dispersal model that allows us to test epidemiological hypotheses. And for this specific epidemic, we could see that for the whole of the epidemic, geographical proximity was the main predictor of dispersal. And that during a specific phase of the outbreak, there was a limited effect of wind on the dispersal. What we also demonstrated is that case-specific local opportunities for spread, as we saw in the previous slide, are efficiently exploited by the virus, and that this results in local networks that are statistically supported by genetic analysis. And really this combination highlights the complex multifactorial nature of avian influenza outbreaks. That leaves me to thank the virology, the sequencing team, and the epidemiological team at Cienzano and Simon Delicourt at the University of Brussels, and to thank you for your attention.